So our final speaker for this part of the symposium is Professor Matthew DeLisa. Dr. DeLisa is the William L. Lewis Professor of Chemical and Biomolecular Engineering at Cornell University. He's also the director of the Cornell Institute of Biotechnology. And his research focuses on understanding and controlling the molecular mechanisms of protein biogenesis. Professor DeLisa has received numerous awards, including election to the American Institute of Medical and Biological Engineering, the American Academy of Microbiology, and the American Association for the Advancement of Science. Today, Dr. DeLisa will be sharing his group's work on cell-free approaches for biomanufacturing therapeutic glycoproteins and conjugate vaccines. It's my pleasure to introduce Dr. Matt DeLisa.
It's a pleasure to be here today, and I want to start by first thanking Kamal and Julian for the invitation to come here and participate in this symposium to kick off the Stack Family Center for Biomanufacturing. I had the great pleasure today, earlier, before coming here, to actually tour the facility, and it made me want to become a student again and go back to hands-on training with equipment of that quality and an opportunity and an environment for making things like the Industry 4.0 for biotech a reality. So we're looking forward to big things from you guys. We're not too far up the road at Cornell University and are very excited in looking for ways to connect with the center here and advance some of the ideas and some of the training and the opportunities, most importantly for students who are entering into this really exciting time in biotech and biopharma. So today, as Julian mentioned, I'm going to spend the time that I have telling you about some work that's been going on in my lab for the last 6 or 7 years on cell-free approaches to biomanufacturing. And the focus today is going to be on attempting to use these systems to make therapeutic glycoproteins and conjugate vaccines. And I want to thank our previous speakers, in particular Ron, for telling us about the current state of industry, where it's headed. And I'd like to hopefully have you leave today realizing that while cells are still the workhorse of the industry, there are opportunities to use the machinery inside of cells in the absence of cellular membranes to develop new manufacturing processes. And hopefully some of the things I'll share with you are an eye towards that future. The vast majority of human drugs that are proteins, so think monoclonal antibodies, but there are many others out there. The vast majority of these are modified with complex carbohydrates known as glycans. And the process by which they're modified is known as glycosylation. And we know these modifications are really, really important for the activity, the stability, the three-dimensional structure, the efficacy of the drugs. And so making drugs that are not just proteins, but that are modified with proper carbohydrate modifications is a really critical part of biomanufacturing. The more complicated drugs get, the more attention we have to pay to these types of modifications. There are two major types of protein glycosylation that you should be familiar with: N-linked glycosylation, the attachment of a complex sugar known as a glycan to an asparagine residue in a protein, and O-linked glycosylation, which is the attachment of a glycan to a serine or threonine residue. Virtually all monoclonal antibodies carry a N-glycan in their Fc domains. In fact, they carry two. And these structures are absolutely crucial for the downstream activity of antibodies in terms of recruiting the immune system and eliminating foreign material and foreign invaders. But there are many other types of glycoproteins out there, and as you can see, they can be quite complicated. So the molecule on the left, that's EPO. This protein is used therapeutically and can have great benefit. And what makes it particularly challenging from a manufacturing perspective is that it's not just a protein, but it has 4 of these glycans attached. 3 of them are of this N-linked variety, and 1 is of the O-linked variety. So these can be quite complicated. And then the last example to the far right are conjugate vaccines. And in fact, some of the most effective vaccines on the planet for dealing with infectious bacteria are these types of conjugate vaccines, which are comprised of a protein carrier that's modified with an oligosaccharide. And that oligosaccharide is derived from the surface of the bacterial pathogen that you're trying to protect against. So these are incredibly robust and protective. So if you're interested in manufacturing a monoclonal antibody and you want to use a CHO cell, the good news is these cells are, in fact, used primarily because they have machinery that introduces these very complicated carbohydrate modifications. If you look under the hood of a CHO cell to try and understand exactly how these cells perform these processes, I don't have time to go into all of the exquisite details, but the takeaway is that it's really, really complicated. There are dozens of enzymes that are involved in this process. It spans multiple subcellular compartments from the cytosol to the endoplasmic reticulum, to the early Golgi, to the late Golgi. And then usually in the case of antibodies and other protein drugs, they're secreted out of the cell. So it's a very complicated process. When it's done right, it is very effective, but very challenging to control and to get it right. And one of the things that we've looked to is, as I mentioned at the very beginning, cell-free technologies. Now, cell-free systems, cell-free protein synthesis in general, is a very long-standing technology. It's been around for a very long time. For making recombinant proteins, it's quite powerful for a number of reasons, and I've highlighted a few of those here. First and foremost, what it allows you to do is to decouple the synthesis of your catalysts. So all of the protein synthesis machinery can be decoupled from their utilization. So you can make all the catalysts when you grow your cells and then prepare the cell-free lysate. But then you can store that lysate and use it at any time in the future. And when you use it in the future, now your only focus is on using those catalysts for carrying out the functions of making protein. So these two important steps can be decoupled. Really what that means is that by moving outside of a cell, you're no longer constrained by a physical or biological barrier, the cell membrane, and you're also no longer subject to cell viability constraints. When we set up these massive stainless steel bioreactors or disposable bioreactors, one of the main challenges is we're trying to keep something alive inside those reactors. And productivity is intimately linked to keeping whatever it is inside that reactor alive and healthy. And oftentimes, what we're asking those cells to do, which is to make massive quantities of our protein products, of our monoclonal antibody, oftentimes asking cells to do that makes cells unhappy, right? And they start to push back. And viability is usually expense, right? That cell death is always something that we're pushing against. So it's a tug of war between making more product, but keeping our catalyst system, our cell, alive. So cell-free systems are powerful because now that constraint is completely lifted. If you've thought about or even done anything with cell-free protein synthesis in your lifetime, you're probably thinking of really small microcentrifuge tubes, which is still where most of the work is being done, including a lot of the experiments I'm going to share with you today. In the laboratory, we're operating in very small volumes. However, in the last, I would say, 10 years or so, there has been a technical renaissance in cell-free systems and many labs coming to operate in this domain. And that renaissance has led to a pretty significant scaling of this technology. There are now several startup companies that are growing and becoming quite successful, Sutro, Biopharma. And these companies are getting these small microcentrifuge tubes of cell-free reactions scaled to 300 liters, even 1,000 liters in some cases. So we're not quite at the 10,000 liters. They can be scaled to quite large systems, despite how you may think of these systems. And they are compatible with a lot of the current infrastructure that we have. They can be operated 300-liter stainless steel reactors. I've seen them operated in wave bioreactor in bags, disposable bags. They're also quite small and nimble. So if you're not trying to produce a protein drug for the entire planet, like Regeneron and others are trying to do in the pandemic, but rather are trying to make a drug for a small patient population or even an individual, this technology, I think, holds tremendous promise for that kind of opportunity. One future of biomanufacturing 4.0 could actually be you walk into a pharmacy and the pharmacist makes the drug for you. Or you're in the hospital and your drug is manufactured in the room next to your hospital bed. And I think a nice thing about cell-free systems is they have an opportunity to provide drug products across these pretty massive length scales in terms of manufacturing. Okay, so a simpler question that we have set out to address over the last few years in my lab is it possible to develop cell-free systems that incorporate these protein glycosylation reactions? So what I didn't say in the previous slide is that the other thing you may know about these cell-free systems is that the vast majority of them, and the ones that have been developed to these very large scales, are generally all based on extracts derived from bacterial cells, in particular E. coli. And as many of you may know, or hopefully if you take nothing else away from today, you will remember that E. coli cells naturally do not perform protein glycosylation. So those really important modifications can't be made by an E. coli host, but also can't be made in most cell-free lysates. So we thought this might be an opportunity for us to make a contribution. We had been working on installing protein glycosylation systems into E. coli bacteria, and that led us to think about perhaps developing lysates from those bacteria that would be enriched in the machinery necessary for installing complex carbohydrates onto proteins. We've tackled this problem at a high level, thinking about it in two distinct stages. So the first stage we're thinking about are the early steps of protein glycosylation. So the initial installation of some complex carbohydrate structure or precursor, or primer, if you will, onto a protein. And if we can succeed in doing that, in a second step, we can carry out in a cell-free environment glycosylation maturation reactions where we can tailor the glycan structures to look exactly like the ones we want that maybe are found on naturally occurring human antibodies, or could be glycoengineered to be structures that afford new properties to the biomolecule, which is a very exciting and fast-growing field. So today what I'm going to tell you about very quickly is on the left, the work that we've been doing towards glycosylation initiation. Take the guts from a E. coli cell that is carrying protein glycosylation machinery, use that to create cell-free extract that will, in most cases, be stored in a microcentrifuge tube and used to drive glycoprotein synthesis simply by the addition of a DNA plasmid that encodes our product of interest. And if you do this right, you drop in your plasmid, you come back in less than a day, and your protein product is made with the desired carbohydrate modification directly on the protein. So your glycoprotein is now made in a very fast and convenient way. So this was work in my lab that was largely the effort of a really amazing graduate student, Tommy Jaro-Antimichai. And so I don't have time to go into the details. The work is published, but he took glycoengineered E. coli in our lab, made lysate from those E. coli, and showed that you could generate a single pot system that was able to make a wide spectrum of different protein products bearing specific carbohydrate or glycan structures. So here are just a few examples, and single-chain antibodies. We've been able to make human EPO that I talked about previously, all with glycan modifications onto these proteins. And in the case of EPO specifically, not only were we able to make these as glycosylated proteins, but we showed in follow-on experiments that the molecules we were making were as active as commercially purchased EPO proteins. We acquired those in a legal way, not from any cyclist. We've done this with N-linked glycosylation. Aravind Natarajan in my lab came along and became interested in O-linked glycosylation. He engineered bacterial cells to carry out this type of protein modification and then adapted it for cell-free systems, and also had similar success demonstrating the attachment of O-linked sugars onto proteins of interest. The other thing that we've been able to do over the work of these two students that I just mentioned is we've been able to install a large array of different oligosaccharide structures onto proteins. And this led us to an interesting observation that one of the oligosaccharides we were able to attach is not the kind you would see on a therapeutic protein, but is one of these surface oligosaccharides that's present any type of bacteria, including bacterial pathogens. And so this led us to ask, in addition to making therapeutic glycoproteins, could we use the same technology to install long-chain oligosaccharides onto proteins as an entirely new route to making conjugate vaccines? And so the general idea here, and this is work that our lab did together with Mike Jewett's group at Northwestern and a really talented student there, Jessica Stark. They had this idea that we could, again, use all of the same concepts that I just shared with you, but what would be really powerful here is that these reaction systems, the lysates you make, can be lyophilized or freeze-dried. So you don't have to use them right away, right? And so then in a freeze-dried format, these can be transported anywhere in the world. And you can do that without the need for a cold chain. So there's an opportunity here to think about entirely new paradigms for vaccine distribution and manufacture that overturns some of the major challenges we have in maintaining the cold chain. And then may even give you an opportunity to think about major decentralized strategies for producing your product, on-demand or point-of-care protein manufacture. So these are some of the ideas that we thought could be addressed with a technology like this. The technology has given rise to a very potent platform for making these conjugates. So another beautiful thing is that this can be a very low-cost technology. These vaccine doses we're making come in at around about $5 per dose, which is still too high if you talk to folks at the Gates Foundation and elsewhere, but we're still way cheaper than the conventional technology, which runs about $200 per dose. So we think we're on the right track to not only creating a new technology, but also one that could be affordable and provide greater access to much needed vaccines. We have seen also, as I showed in the EPO example, that not only can we make these things, but they're efficacious. And this is an animal study that we did with some of these cell-free derived vaccine candidates, showing that they are able to completely protect mouse from pathogen challenge, where the pathogen that was used in this experiment corresponded to the oligosaccharide structure that was attached to our protein carrier conjugate. So these are the two platforms. I told you about the one on the left. I don't have time today to tell you about how we take primer glycans that are initially installed on proteins and then turn them into whatever structures we want. But there's a huge opportunity to elaborate these glycans, opening up new opportunities for glycoengineering, which is basically a way of making biosuperior drugs because the carbohydrate structures themselves have been tweaked or modified to give a better therapeutic effect, something we're very interested in. We think there's pretty massive opportunity for cell-free technology, not to replace any of the things we've heard about today or any of the things that are going on here in the Biomanufacturing Center, but to operate alongside and provide alternatives. And that's really what we need, I think, as an industry, right, is to have lots of different shots on goal with lots of different technologies that can all be brought to bear in making the medicines that we need going forward. So hopefully the next time I come to the center, we'll have some cell-free systems that are up and running in those same bioreactors. And if not, I'll do my best to try and make that happen. So with that, let me thank first all of the great individuals, both students, postdocs, and collaborators that I've had the great fortune to work with on some of the things that I shared with you here today, the funding agencies that made a lot of the work possible. Thank everybody for your attention, and if there's time for a question or two, I'd be happy to take those.
One of the questions that I would have is timeline. How long does it take from inception to having a working vaccine? Not necessarily through all the health authorities, not counting that.
Yeah, so great question. So broadly speaking, and not just for the conjugate vaccines, we think one of the real game-changing aspects of this cell-free technology is that when you have the lysates prepared and stored and ready to go, the timeline for making drug products. So you mentioned this in your talk about there's really a key race to get from discovery, early stage discovery, and getting small doses to the clinic, right? We think that's a real opportunity here with the technology. It's quite fast. From the time you identify a pathogen for a conjugate vaccine, it's quite fast because you actually clone the genetic pathway that encodes the oligosaccharide biosynthesis. So that's a day or two. You put it into the system probably takes less than a day, but let's just call it a day, 2 days max to make your product, depending on how much you're trying to make. So you could conceivably, we've done back of the envelope estimates, get this done in about a week if you're really turbocharged and you know what you're doing. And so with that in mind, we've been approached about pandemic response, right? And not just thinking about coronavirus, but all kinds of outbreaks that we've dealt with over the last any number of years. And so a technology like this, we think, has that opportunity. And the good news is you never actually have to handle the pathogen. Current conjugate vaccines, for example, you're handling the pathogen to shear off or remove those oligosaccharides. We never have to touch those. Everything is completely synthetic or biosynthetic. So it also, we think, creates a much safer work environment with less hazards in terms of material handling.